All right, folks, we're going to get ready to start session 6, From Concept to Clinic, the emerging role of precision immunotherapy in kidney cancer. Our moderators are Dr. Hans Hammers and Dr. David Braun. And I'll let you take over.
Hey, good to see you. Thanks for doing this.
Great. You can sit right there. You can, I think, one over. Very good. I think you have to go one over. Yeah. Great. Welcome, everybody. So I'm here with my very talented co-chair, Dr. David Braun from Yale, Hans Hammers from Southwestern. And we have really experts in their field to show kind of like where we are, where the field might be going. And so without further ado, we will start with Dr. Chuck Drake, a colleague of mine, formerly from Hopkins, went to Columbia, a very accomplished physician scientist, went into industry, and now with Johnson & Johnson. So thank you very much.
You know, thank you for the invitation. I don't get out much, and an invitation like this allows me to get updated with kidney cancer and to see all my old friends. These are my disclosures. The top disclosure is the most relevant. I work for an evil drug company known as Johnson & Johnson Innovative Medicine. Johnson & Johnson works on bladder cancer, prostate cancer, and lung cancer. We do not have an official program in kidney cancer, but I'm trying to change that. The other thing is, it was really interesting to see that people are still interested in LAG-3. I'm a co-inventor on some of the original IP with Jonathan Powell, who will speak later. So, what is the future of IO in solid tumors? Not just kidney cancer, solid tumors in general. And I can tell you, for a long time, I thought the top line was the answer. I thought it was going to be combined checkpoint because it's synergistic in mice and we were going to cure people and life was going to be great. And frankly, although we see additive activity, we've not really ever seen the synergistic activity with any of the combinations, LAG-3, TIM-3, TIGIT, any of these things. Autologous CAR has promise, but it's a little bit logistically challenging. Naomi Haas is leading some of these efforts, both in prostate cancer. We're looking forward to data. And you're all aware that a company called Allogene has shown some pretty nice data with a CD70 allogeneic CAR T in kidney cancer, of course, with some toxicity. My friend Laurence asked a brilliant question. She was like, what about ADCs, man? I'm like, okay, that's cool. I mean, pharma companies love ADCs. I mean, love. And you can tell this because they made big deals, right? My group is developing ADCs for prostate cancer, and I just told my friend Laurence, this is not easy, right? It's a nonlinear problem with multiple variables that's basically reasonably, actually it's not reasonably, it's totally empirical right now. So I'm going to make a little bit of argument. I used to have question marks. Is it going to be T-cell engagers? Is it going to be T-cell engagers? At least I think the prostate cancer data tell us that it could be T-cell engagers. So what are these drugs? So basically, they're bispecific antibodies. They come in multiple flavors, shapes, and sizes. I used to have a slide, I actually had to take it out for time, about the different flavors, but this is basically one that's FDA approved. It's a GPRC5D for multiple myeloma. People talk about these wrong, okay? What they say is, oh, well, one arm binds to the tumor, the other arm binds to T cells, and it drags it over. Excellent. That's not true, okay? What happens is the arm that binds to T cells binds to the CD3 epsilon chain, and it sends strong signal 1 and some signal 2 to activate the T cells. And how do you know this? You know this because you get cytokine release syndrome. So patients develop side effects that show that their T cells have been activated. Second of all, you see rapid activity in multiple tumor types, and I'll show you some data from prostate cancer. I believed this when I went to Columbia, okay? So I went to Columbia in 2016, and I'm like, we are going to treat prostate cancer with T-cell engagers, and we're going to open every trial we can. And I had a great team there, great people, Jess Hawley, many other folks, Mark Stein, now Carrie Runcie, who spoke before. And we opened 3 trials, okay, for prostate cancer, all directed against PSMA, and they all failed. Failed. Like either they were toxic or they didn't work. Regeneron is still open. They're moving along with the CD28, but it's a very challenging drug. So the point that I'm making with this slide is these are not easy drugs. It takes multiple shots on goal. This was Johnson & Johnson's first shot on goal. It's a PSMA. The one that I'm going to show you later is not our second. It's not our third. It's our 4th shot on goal. And it took many cohorts of optimization to get the dose and schedule right. But this is what happened, okay? So I was at ESMO in 2023. This paper came out a little bit later, 2024. And there was a presentation from data we had never seen before. And all the Johnson & Johnson prostate guy goes to the session. And Kevin Kelly is going to present this new CD3 redirector for CD3 T-cell engager for prostate cancer. It's against STEAP1, which is not the cleanest of targets. And we were all sitting there and like this just blew us away, right? So basically these data show responses of immunotherapy in a cold tumor known as prostate cancer. Deep responses, Kevin showed that they're durable, and actually occurs across multiple disease settings, and it was just amazing. What this shows is that in prostate cancer, a cold tumor, T cells can kill tumor cells. It's amazing. The other thing is that prostate tumors are not really infiltrated, unlike you heard from my friend Dave this morning that kidney cancer cells are. So the cells that are working here probably are not the ones in the tumor. They probably have to get dragged in from the periphery. These drugs are not without challenges, actually. So these drugs mostly cause cytokine release syndrome. This is not like in the kidney cancer doctor wheel of treatment for the most part. This is what like heme doctors work on: hypotension, hypoxia, tachycardia, many things, usually treated rapidly with TOSI. Many of these drugs need to be given as an inpatient, okay? Or at least with a long observation period. And this is the adverse event profile. A grade 2, just for reference, a grade 2 CRS is bad. You're supposed to have an ICU bed ready if you have a grade 2 CRS. And you're supposed to have TOSI ready. So these are powerful drugs, both in side effects and activity. And in activity, this is also amazing for immunotherapy. This drug shows activity in liver mets. And so the bottom line is that I think this experience, it's AMG 509, zolaritomab, taught us or taught me that T-cell engagers, well, probably taught everybody, that T-cell engagers can have activity within solid tumors. And there's a beautiful cancer discovery paper, and this is now in phase 3, and I wish them nothing but good luck. It's an important drug. What we have done actually is found a little bit better of a target antigen, and we did not do this ourselves. We did this with the help of colleagues from a place called Memorial Sloan Kettering. So Hans Lilja, Dave Ulmer, and Dan Thorak had a small company called Diaprost. And their idea was to diagnose prostate cancer, diagnose and diagnose only. They developed an antibody to an antigen called KLK2. And their antibody is really interesting because KLK2 is a secreted protein, and this antibody binds as it's being secreted. This antibody was probably lucky on their part. Johnson & Johnson, I played a little bit of a role, helped in-license this as a target for prostate cancer. What we need for kidney cancer is we need a KLK2, okay? If we had an antigen that was this clean, we probably could develop a successful TCE without too much difficulty. The other antigens of prostate cancer, by contrast, are fairly not clean. So PSMA, as you know, is expressed in vasculature. It's expressed in some neural cells. It's expressed in the kidney. And STEAP1 that's the one we were just talking about. STEAP1 is a target. It's pretty good, but you can see on below, it's both expressed in multiple normal tissues. So this is a slide that was presented at ASCO by Cappuccino Baldini and recently published in a JCO paper showing the KLK2 CD3 engager that we have. I want to make 2 points. So this is not, again, this is not our first rodeo in CD3 redirectors for prostate cancer. It's our 4th. And these drugs are not easy to work out the dose and schedule. So it's hidden in the paper, and we don't say it loudly, but I'll say it here. This was cohort number 22, okay, in the phase 1 trial, number 22. So it takes a long time to work out the dose and schedule. This drug is given IV, not sub-Q. It's given IV Q6 weeks. The target dose of the recommended phase 3 dose is 300 mg per kg. And you can see on your right the step-up dosing. So this is how it works. So you get a little baby dose. I think it's 3.5. Then the next week you get a little bit bigger dose, still a baby dose, 18. Then you get the full treatment dose. These first 3 doses are given with steroids, okay? A pretty good slug of Dex, I remember it's around 16 or so. And then after that, you don't have to give any steroids. It's amazing, actually. This drug is well tolerated. I took out that slide, but the bottom line is we saw no CRS grade 2, 0. CRS grade 1 of about 5%. So this is an outpatient drug. And it works, actually. I mean, this is the PSA response curves. It's about a 43% PSA response rate. And this allows it to be developed in late-stage prostate cancer. But more importantly, and you guys know this better than anyone else, immunotherapy, PD-1 blockade, for example, is a monotherapy, is not what you want. You want a drug that you can combine. And this kind of a response rate plus the safety profile will allow us to combine this with other drugs in prostate cancer. Okay, so the bottom line, honestly, CD3 redirectors work in a cold tumor called prostate cancer. I showed you 2. There's actually several other ones in development. What about kidney cancer? You heard yesterday from Ritesh, actually, from Memorial, about a drug against ENPP3. He didn't have time to talk about this, but I will. So ENPP3 is a pretty good target for kidney cancer. It's what we call a polarized antigen. So it's expressed primarily on the apical surface, and the theory, the hope, the dream is that in normal tissues, antibodies don't get to that apical surface, right? And so that when the architecture is disrupted, as in kidney cancer, then the antibodies can traffic to the tumor. Just to show you the expression, it's actually amazing. So like 93% of clear cell expresses this. We presented, my friend Smriti presented this at ACR in 2024, so this is public, but it's the expression across kidney cancer, especially clear cell, is quite good. I told you before, right? I'm at Johnson & Johnson, we don't have a kidney cancer, so why are we doing this, you might ask? Well, it's also expressed in lung cancer and in colorectal cancer. We do have a lung cancer effort. You can see how for kidney cancer, you probably don't need patient selection, but for other tumor types, you probably do, okay? So that's all I'll say about that. This is an important part, okay? These drugs are not all the same. So if the Xencor antibody works and ours doesn't, would I be surprised? No. If ours works and theirs doesn't, would I be surprised? No. I really hope that they both work, actually, but what I can tell you is that the models that we use to develop these antibodies are these, I'll use a Trumpian word, they're rigged, right? So basically what you do is you mix tumor cells, and you mix T cells, and then you mix the antibody together in a little dish, and guess what? The T cells are right next to the tumor, and this can drag them over, activate them, and you can kill, actually. So you can use this to rank them, but it doesn't really, probably the best way to predict. Our drug is a little bit different. We have a SCFV for the CD3. The one thing that's a little bit in design principles understood is that if you have a lower affinity CD3, you might have less CRS. And so ours is designed with a lower affinity, and that's the NP3 binder. So take home, I saw Hans looking at his watch. So bottom line is that CD3 T-cell engagers work in cold tumors and in prostate cancer, right? So there's no reason why they shouldn't work in kidney cancer. But it is a matter of iteration. There have been 20 PSMA CD3s, 20, okay? And now they're finally starting to work. It just takes iterations. Molecular design principles, we don't know. But the CRS is a real thing. It's addressable with TOSI and careful management. It would be better if you had less. And again, the CD3 ENPP3 is in phase 1. Maybe next year we'll be able to present some of the clinical data, but it's still in one of those dose escalations with many cohorts. I don't think we're up to 22 yet, but we are in multiple cohorts. And that trial is being run by one of our former Hopkins colleagues as well, Katie Thornton. So with that, I'd like to thank you for your attention and turn it over to Hans.
Great presentation. We have a few minutes for questions. Dr. Atkins?
Yeah, I'm supposed to be moderating, but I'll start with a couple of questions. So typically when we're giving immune therapy, we don't want to have steroids at the beginning. So it's interesting that you can get away with giving steroids to address the toxicity. And what do you think is responsible for the tachyphylaxis that you're seeing? We saw something like that in our IL-12 studies. We never fully worked that out, but what's going on with the T-cell engagers?
Question. So the immunological mechanism by which these step-up doses result in tachyphylaxis accommodation to the drug is really not known, actually. And so one theory is that maybe that you have these pre-existing T cells that you, those are responsible for the initial CRS, and maybe with the steroid you actually energize or turn those off. That's the theory. To be honest with you, we have FDA-approved drugs, right? So Tec-Tau for multiple myeloma, it has not been proven, okay? So the mechanism by which initial treatment mediates this tolerance is not known. And the steroids, I gotta tell you, I agree with you. It's just stunning to me that you can give steroids with an immune therapy and then develop a sort of a regimen where eventually you don't give it. Yeah, what those steroids do in the beginning, those steroids actually almost certainly, they facilitate the tachyphylactic process, which is still not understood. How's that?
Yeah. So,
probably pretty unsatisfying, actually.
Yeah. So
I'm not satisfied either.
A second question I had, the T-cell engagers I know about in uveal melanoma are against internal antigens, and so they're HLA restricted. What can you comment about potential antigens in kidney cancer that would not be surface expressed?
Yeah, it's a great question, and Mike, thanks for bringing it up. As I said a little bit during the talk, like most of the basic principles around these drugs are reasonably well known, how to arrange the T-cell engager and how to screen them. The source of antigen being an intercellular antigen that's HLA restricted, as we see in uveal melanoma, and actually it's being developed in lung cancer and several others. So if there are good targets for which we can develop binders, we can make a drug. In terms of the optimal antigen, like we have a world full of profiling people, like, you know what I'm saying? So if you guys come up with a target antigen, we could think about working together to make a drug.
Dr. Bergerol.
I was surprised what you said about the T-cell engagers, that they are being evaluated in vitro just by mixing lymphocytes and tumor cells. I assume these are autologous, and I wonder what would you think about leveraging autologous PDX models where you are reconstituting the PDX with autologous PBMCs from the patient? Would that take it a step up, or you think it would be the same thing?
Oh, you give us too much credit, my friend. So these are not. These are allogeneic. It's a tumor cell line, somebody else's PBMCs, a donor that we buy, and the T-cell engager. In some groups, in some models, people pre-activate the T-cells to even rig the system further. I think that you're correct, actually. Using PDXs or an autologous system probably could lead to a different level of, actually, a finer level of discrimination for sure, but that's not how these studies are currently done. Not only not by us, but really by across the field, actually. If you look at T-cell engagers, that in vitro experiment I showed briefly at the end, that's a standard experiment to rank your T-cell engagers.
Thank you.
I think we have time for one more question, Dr. Tanier, and then we have to move on to the next speaker.
Two questions, quickly, brief one. First, schedule. Is it weekly? Because if it's weekly, can it be, instead of weekly, can we do every 3 weeks? Because really, practically, you're not going to be able to prescribe something like this to patients every week, especially if you admit them to the hospital. So that's what. Secondly, is CD3, CD28 engagers better? Are you doing that, or anybody else is doing that?
You're like a brilliant man, my friend. So two answers. So first of all, this drug was originally developed every 2 weeks. And the person who was leading the study, an MD PhD named Victor Villalobos, noticed that if we took a break, if a patient had a toxicity, and then we retreated much, much later, there was a better response, actually. So we tested 2 weeks, 3 weeks, and 6 weeks, and this drug is given, check it out, every 6 weeks, IV. And we have translational research data that we hope to present at ESMO, showing that giving these drugs too often almost certainly causes exhaustion. Okay, so and actually in the real world, I mean, Teca's the multiple myeloma is approved every 2 weeks, but there's some off-label use where people are giving it a little less frequently, and that's actually a great point, by the way. So these drugs, people just bring it in like, oh, every week, oh, every 2 weeks. It totally matters, and that's another part of why it takes a while to develop the dose and the dose and schedule. And then finally, sure, I mean, so you want signal 2, right? Right? I mean, for T-cell activation, you need signal 2. So we have another drug, a PSMA CD28, that's in a phase 1 study with this drug. So it's KLK2 CD3 plus PSMA CD28, and that's, guess what, in dose escalation. It just takes a long time.
Great. Thank you so much. Very great presentation. The next speaker is another very dear colleague of mine, Dr. Rosa Nadal Rios. We were at Hopkins and she went to the NCI. She's very well known for her TCR therapy that she directed there, and then she was recently recruited to the Fred Hutch, and she will talk to us about novel targets for cellular therapies in renal cell. Thank you so much.
Thank you very much, Hans. Let me see if I can learn how to go on that. So I have been tasked to talk about novel targets for cellular therapy in renal cell carcinoma, and there are 3 big groups of immunogenic targets that has been recognized in cancer, tumor-associated antigens that are encoded by the mutated genes and can be expressed also in normal tissues, in selected normal tissues, have high structural similarities with normal proteins. Targeting these antigens has led to severe toxicity in the past, but they are highly attractive because for off-the-shelf immunotherapies, because they are shared among patients and also across malignancies. I have listed some of these tumor-associated antigens that we have experienced in red, the ones that are relevant for clear cell renal cell carcinoma. The second group are tumor-specific antigens. They are the result of genomic perturbations that occur exclusively in tumors. They are rarely shared among patients and have been, because they are intracellular proteins that have been commonly treated with TCR based chemotherapy, based cellular therapies in the context of HLA restrictions, limiting further the applicability in large populations of this type of therapy. So targeting these tumor-specific antigens required very, very highly personalized therapies. This is, and we don't have any, I want to go back. So in the last column, as you can see, I have not listed any tumor-specific antigen in red because there is not a single tumor-specific antigen that has been validated as a target for cellular therapy, and we have not tested them in clinical trials. This is probably the result that we don't have a very good understanding of the full antigenic landscape in kidney cancer that is derived from the common molecular alterations. We don't know for sure what immunogenic tumor-specific antigens are recognized by the adaptive immune system and lead to the immune attack of the cancer. The few neoantigens that has been described, for example, some described from transplant patients by the Catacodis group, or some derived from immunogenic human endogenous retrovirus. They have not gone through the functional validation. The TCRs has not been discovered, and this has enabled the redirection of T cells, the engineering of the T cells against these tumor-specific antigens. Most of the cellular therapies that are currently clinically evaluated, targeting antigens in clear cell renal cell carcinoma, basically are CAR-T cell products that target overexpressed antigens that are mostly the result of transcriptional activation of HIVs in the tumor. Except the exception to this consideration is human endogenous retrovirus E. So this is a very busy slide with a summary of all the cellular therapy products that has been and are currently being tested in clear cell renal kidney cancer. One allows the enrollment of papillary renal cell tumors. Highlight few targets, CA9 that was reviewed yesterday by Dr. Marasco, and all the efforts that his team has done to overcome the initial toxicities that were observed with this, with CAR-T cells targeting this antigen. CD70, there has already been 50 patients treated in the Transverse trial. This is the largest experience that we have with cellular therapy in clear renal cell carcinoma, showing clinical activity and also acceptable toxicity. The investigators are also gaining experience on the relationship of the expression of the target responses and how the CAR-Ts, the CD70 CAR-T cell eliminates CD70 positive T cells from the recipient, that it does not seem that has clinical repercussion. Many other CD70 targeting agents are entering this clinical evaluation. HERVE that I will review briefly, and other targets like MAC1 or, for example, CMET, that are basket trials, allow the enrollment of clear cell renal cell carcinoma patients, but these are overexpressed antigens expressed across different malignancies and not specific for kidney cancer. Two products that enter clinical investigation, 2 trials targeting AXL and ROR2 have been terminated, and it's unclear, it's not reported the reasons why. So I wanted to take a moment to review, to comment on TIL therapy. because it is a form of adoptive cell transfer that has not been largely evaluated in kidney cancer, mainly because the historical difficulties in expanding, ex vivo expansion of TIL from the patients. The initial trials show up to a 40% of TIL production failure rate. But I have seen that some of the groups are working and are publishing on new forms of new manufacturing protocols to efficiently expand TILs in clear cell renal cell carcinoma. And I think there is an opportunity because TIL therapy can be highly customized based on the neoantigen recognition in the autologous tumor. That can be a highly personalized therapy. Also, comment on this small trial, a small study that this group screened TILs against more than 1,500 neoantigens that are derived from point mutations and also indels. And they replicate what we know from other epithelial malignancies that are very many neoantigens presented in the HLA system, but very few are recognized by the immune system. Most of them are private neoantigens, and in this small group of patients, none of them were derived from known shared mutations. I see opportunities to learn more about these tumor-specific antigens in kidney cancer and other malignancies that has a low tumor mutational burden. Through this profiling of TILs, some groups has been able to develop both TIL therapy and also TCR-based therapy targeting these tumor-specific antigens derived from exomic alterations. And the last part of the talk, I'm going to focus on the human endogenous retrovirus E, the difficulties of the discovery and targeting of this immunogenic antigen, because it's the one that I know the best. So here, HER-VE was discovered after the isolation of a cytotoxic T-cell clone for a patient that underwent a haploidentical stem cell transplant and had a complete response. This cytotoxic T-cell clone, it was CD8, was recognizing the tumor but not normal tissues, the autologous tumor of the patient, but not other normal tissues or other targets, and was also recognizing other tumor-derived clear cell renal cell carcinoma cell lines in the context of HLA-11 restriction. The investigators did an awesome job with the screening of all the transcriptome of that tumor. So it was a very comprehensive screening that allowed the identification of the transcripts that were derived from a human endogenous retrovirus E that was not supposed to be transcribed in human cells. So I just wanted to highlight the level of confidence and validation that it requires to identify one of these targets when you don't know exactly what you are looking for. For those that are less familiar with the concept of human endogenous retrovirus, they are remains of viral infections to our ancestors that has integrated the genome in the germline and has been transmitted to the offspring. Have been silenced, these human endogenous retroviruses during the evolution through mutations and also through promoter methylation. In the case of clear cell renal cell carcinoma, and specifically HER-VE, not other human endogenous retrovirus, the promoter of this provirus is hypomethylated, allowing the interaction of HIF2 alpha and the transcription of this provirus. It is selectively expressed as a consequence only in clear cell renal cell carcinoma, and in normal cells the promoter is hypermethylated and this provirus is not transcribed. This makes an immunogenic target very, very appealing for cellular therapy targeting, for redirecting the engineering T cells and redirecting this immune system to the recognition and these peptides in the cancer cells. So the TCR was was sequenced and encoded in a retroviral vector, and these cells were transduced. And the next steps was the validation of this HER-VE transduced T cell for a specific recognition of this peptide in kidney cancer cell lines. So this experiment showed that there is a specific recognition not only of the peptide, but in the HLA-11 restriction. This is the kind of validation of many of the neoantigens, or tumor-specific antigens, that has been described in kidney cancer. Has not the discovery of the TCR functional assays has not undergone, and that is why we lack clinical trials targeting these tumor-associated antigens. The investigational plan, we scale up the production of cells, the investigation, and we conducted a phase 1 trial. The investigational plan was very, very standard for cellular therapy, lymphodepleting chemotherapy, infusion of 1 dose of cells, and then interleukin-2. We treated 15 patients. We screened 185 patients to identify only 15 patients that were HLA-11 and were treated in this clinical trial. We didn't see any dose-limiting toxicity. We have 2 manufacturing failures. Unfortunately, after all this validation and all this hard work of the team, the clinical activity was very limited. Only 1 patient had a partial response that was very short-lived. Looking at the in vivo HER-VE TCR T-cell kinetics, we only see 1 patient that expanded a very huge clone, but the rest of the patients had only low to moderate detections of these cells in the circulations, and only during the administration of interleukin-2. After that, these clones were detected only at later points at very low levels. This low level of expansion and persistence probably has limited the clinical activity of this product. The team has done very, very comprehensive work to try to understand why this happened by analyzing the infusion products for their composition through a multi-omic approach with single-cell RNA sequencing, metabolomics, flow cytometry, and the results, the data that has been completed and has been submitted for consideration to presentation at SITSIC. The future directions for this target. So HLA-11 restriction is a target that is a restriction that is mainly it is highly prevalent in Asian populations, but not in Caucasian. So there is another predicted peptide that is HLA-2 restricted, that is immunogenic and only expressed in the tumor, is cancer restricted, that is undergoing the validation to develop a cellular therapy against this target. The transcripts that encode for the envelope of this virus are also restricted to the tumor, and these have the potential for future development of CAR T cells. The identification of new peptides with new RNA techniques is also one of the aims. Obviously, manufacturing improvement, because it seems that the product was very highly differentiated, without capabilities for proliferation and persistence, and that is a very important part of the development of these cellular products. As many of them, they will fail, and it will be because the product is not potent enough. And then development of combination therapy. This work was done at the National Institute of Health, at the government, by Dr. Charles teams, because it has been like teams over 20 years working on this project. And I'm going to finish here my talk. Thank you.
Thank you so much.
Thank you so much. Maybe 1 or 2 questions? Any questions?
I think Jim?
So great talk. Is it possible that the HER-VE expression, also the HER-VE expression in the tumors, was one of the reasons why there was such low activity.
Okay, yeah, I got you now, sorry. So it might be, so the expression of HER2 is variable and it's dependable not only on the the hypomethylation of the promoter, but also probably the levels of HIF2 alpha. And in the preclinical development of the target, it has been seen that those cell lines that have higher expression, not only of HER2, but also HLA-11 molecules, are the cell lines that are more susceptible to the killing by these transduced HER2 T cells, HER2 TCR T cells. So I think this is one of the considerations. I think the data that the lack of expansion in most of the tumors, even in 6 patients that were treated at the last dose level, I think there is a big component of the way the product was manufactured. It was manufactured in a very like traditional, I want to say not all, procedure where the rapid expansion program is very prolonged. It lasts 14 days on interleukin-2 and 15 and allogenic feeders in the culture. So we have the suspicion that might be different factors that have impacted the lack of activity, but probably one of the that you can address is the manufacturing of the product. There has been also a lot of investigations on how to upregulate expressions on HER2 in those tumors that maybe the promoter is also only partially hypomethylated. So these are questions that are not, I don't have an answer, but are the focus of a lot of the research that is being done on this target.
Great. Great presentation. In the interest of time, we're a little bit over, a little bit behind. We're moving on to the next speaker.
Perfect. And it's our pleasure to introduce Sidi Chen, who's really a star genetic engineer based at Yale. And so his work has really used systematic genome-wide approaches to identify how to make T cells fitter and more functional with applications in cell therapy and beyond. And he's already done some work in kidney cancer, but I think one of the secret goals is to lure him into kidney cancer a bit more, because I think I'll have wide applications. So thanks so much, Sidi. Yep, it should be.
No?
They'll adjust. Yeah.
Okay. So I'd like to thank David and all the organizers for inviting me for this meeting. And today I'd like to give you a flavor of what we do. Dr. Drake and others already mentioned there's so many good drugs for the field, including monoclonals, ADCs, and bispecific T-cell engagers. And why do we still need cell therapy? What I'm saying here is that cell therapy is such a successful drug in hematological malignancies, but in solid tumors, particularly CAR T, there's so many trials, but the majority of them have yet to prove to be effective. And this is because we believe that our body is designed or evolved for so many years to achieve a balance of immune activation and immune silencing. Because if immune system is overactivated, we have all the immune diseases. If immune systems are not activated enough, then we have problem with cancer, infection, etc. So the T cells are surveying our body to achieve this balance, to fight all kinds of diseases and maintain homeostasis. And the problem with the current drug, no offense, that with the ADCs and T-cell engagers, that they rely on the wild-type T cells, which is the T cell that reside in our body, which is without genetic modifications. But we need T cell to work even better in order to achieve the efficacy in solid tumors such as the kidney cancer. And but there's so many genes in our genome. How do we know which gene we can modify to make them better? The T cells genomes are controlled by a lot of regulatory network, and we call those the intracellular immune checkpoints. So unlike the immune checkpoint on the surface, like PD-1, LAG3, CTLA-4, and the T cells are not only regulated by the surface checkpoint, but also intracellular checkpoints, which block the T cell from being overactivated. So by modifying one or multiple of those checkpoints, we can potentially make T cells a lot better. But then how do we do it? There's so many of them, and we decided to do it in the shotgun approach using large-scale screens. So we developed a system called CLASH, which is called is a CRISPR-based approach utilizing pool engineering of adeno-associated virus, so we can modify the genome of T cells in a massive parallel fashion. So in this fashion, we can create many, many different drug candidates, like each of the mutants being a distinct CAR T cells, and then we can make tens of thousands of them, and we can subject them to natural selection, either in vitro or in vivo. So this is an example of how we select those mutants. Like in this case, we create several thousand of the CAR T cells, each one being a distinct genetic modifier. So many of those mutants are subjected to the race against cancer cells. We feed cancer cells over and over again, and then the T cell can fight cancer cells. Some of them die, some of them prevail. And at the end of the day, as you see from the bottom towards the top, and after 7 to 8 rounds, we're ending up with a couple dozen winners, which can outlast the cancer cells. Because the majority of T cells undergo T cell exhaustion, which is common in the solid tumor. And later on, we figure that even if we knock out the best winner in the single mutant, are still not enough. This is T cells regulated by a complex genetic network. And then we decided to make it even more complicated, is that by modifying 2 genes at a time. So, but how do you know which 2 to modify? Then we design a combinatorial screen approach, which is a CRISPR dual perturbation screen in which we knock out 2 genes at the same time. So we design a library targeting 71 genes, all of the 71 genes combinations, which is like 2,500 combinations. And then we have redundancy, have 16 different guide RNAs or individual constructs targeting each of those pairs. So that's several thousand of the 30,000 combinations. And then we also knock out single genes as controls, as well as the dual NTC or dual neutral control. So by perturbing roughly 50,000 perturbations, we are making dual perturbed T cells. And we are testing those in a pool fashion, putting these 50,000 mutants, each one having either 1 gene or 2 genes being knocked out in the CAR T setting. So we test these 50,000 different CAR Ts and see which ones of those are being the most superior. So in this screen, we identify roughly a couple dozen of those pairs which make T cells much better than the control or the wild type, which is the natural T cells in our body. And one of the top hit we found is called NR41 SOX3, and one is being a transcription suppressor, and the other one is a cytokine suppressor. So this is a 1-2 punch. By first releasing the cytokine regulator and then releasing the transcription regulator, we can achieve a much better efficacy. And this is still preclinical, but I want to show you the data is that in these tumor growth curves, you can see from the bottom left, if you don't treat a tumor grow, of course, and then if you treat it with CAR T, this is a solid tumor, and you can see there's a minimum or marginal efficacy, which is totally expected. So, and then you may guess, let me knock out PD-1 and CTLA-4, and that only adds to a small margin of the benefit to the CAR T, so you can randomly guess. And if you knock out either one of the single genes, either NR41 or SOX3, you also add a small benefit. Only when you knock out both genes simultaneously in those CAR T cells, you achieve a superior in vivo efficacy in these preclinical models. We have quite a few of them, including PDX, CDX, multiple cancer model, and this is always the case. So we believe we found a gene pair that's very potent and very superior in turning the wild type T cell into non-exhausted T cells. And we did a lot of work to dissect the mechanism of why this pair is so good. And we did a single cell RNA sequencing profiling the T cells, the CAR T cells from in vivo. And what we found is that these T cells have more potent effector function and also a more immune memory effect. And we also dissect the pathway, and what we found here is that the mTOR pathway in the DKO is being upregulated in the memory population, and that's also associated with the NF-κB pathway and interferon gamma TNF alpha pathway upregulation. So I will skip for the downregulation part. And when we look closer under the microscope, this is electronic microscope, and what we found is that these dual perturbed T cells have a lot more mitochondria than the control T cells as well as the single knockout counterparts. And so you may wonder, these T cells are so potent, but are they associated with a higher risk of toxicity? And then we did in vivo a preclinical tox experiment to look for the tissue pathology. And what we found is that there's no significant difference between the single knockout or the double knockout CAR T cells as compared to the control wild-type CAR T cells. And also the serum cytokine level maintained roughly the same between all the different groups. And then we also did a whole genome sequencing to see if the gene editing have created any undesired off-target translocations or off-target editing, and we didn't find any upregulated level of those off-target events. So which means these T cells have provided a superior potency benefit, but without substantially increasing the toxicity risk, at least at the preclinical level. And also I want to echo Dr. Drake's talk again, and we independently found ENPP3 as an emerging target as kidney cancer, and like roughly 4 or 5 years ago, and then that also funded by the DoD KCRP award. And we also realized there are ENPP3 ADCs and T-cell engagers out there in the clinic in phase 1 early phase trials. They're safe, which is good news, but they're not very efficacious. We're seeing single-digit overall response. So we are now developing ENPP3 CAR T therapy, and we have a fully human drug candidate that's targeting ENPP3 and sub-nanomolar level affinity, that we have the CAR T cells being genetically engineered as well. So we also see strong efficacy in mice. For the sake of time, we're not going to go into detail of those. So we're planning for a clinical trial as licensed to a company. Hopefully we can see some clinical data later. So with that, I'd like to thank the group that did the work, and again the KCIS for the opportunity to present our work. Thank you very much.
Quick question for you, my friend. So that was beautiful, actually. So you know, ENPP3 in mice and humans are different, actually. So did you have to make a CAR T for mouse ENPP3 to test for mouse toxicity?
Yeah, that's a great question. We actually made both, and of course, the drug candidate is a human version, and the surrogate is a mouse version.
And you tested the mouse version in the mouse and didn't see too much.
Mouse in the mouse and human in the xenograft, of course.
Fantastic.
Yeah.
Thank you.
So I noticed in your xenograph experiments, the size of the tumor was about 50 or so cubic millimeters. Is size important for these experiments?
Yeah, absolutely. So that's a great question. In that experiment, the size is relatively small, about 50, and then we have PDX when we let it go to 100. And more exciting recently, we have a model when we let it grow to 400, and then that's an in vivo CAR, which is a virus, and then we inject the human PBMCs, followed by a virus injection, which may in vivo in situ CAR T, and that wipe out the tumor that's 400 in all the mice.
Thank you.
Thank you.
Using the genes that, the pairs of genes that seem to be particularly effective, and using that to select or limit the number of targets, did you do any experiments looking at 3 perturbations to see if that was any better than 2?
Okay, great question. So knocking out 3 genes we believe would potentially increase the chromosomes translocation concern. So instead of knocking out 3, we are knocking out 2, but overexpressing another, and that the overexpression can be built into a CAR construct, so we're not making manufacturing more complicated. So that's a great question. So.
Jonathan.
So there have been a whole bunch of screens similar to what you did, but in our endogenous T cells and T cells. And what was curious to me is SOX3 and the other gene are not like the fan favorites of what come up in those screens. And I'm curious if you think that what you found is more specific for a CAR T cell than a regular T cell, or what are your thoughts along those lines?
Yeah, thank you. We in fact did the first in vivo screen, and what we up to today, all the other screens, including our own, are single knockout screens. And you're going to see many, many different genes in many, many different contexts. And what we are presenting here is a double knockout screen, which knocks out 2 genes at the same time. And that's why you're seeing all the weird combinations you can never guess, like for example, SOX3 and NF4A1. So because there's a synergistic effect of one gene and another gene. In this case, the first gene is a cytokine suppressor. If you knock it out, the T cell being highly active, but they can only be active for a while because then they become exhausted. Only when you knock out the second gene, you make them last a lot longer. So that's a 1-2 punch effect.
So does this 1-2 punch effect work in normal T cells? Have you tested that?
We've so far tested only in CAR T cells, but different type of CAR T cells. Yeah.
That's great. Thank you very much.
Thank you.
Perfect. And so next up, it's my pleasure to introduce Patrick Ott. Please forgive him, he's actually not a kidney cancer doctor, he's a melanoma doctor, but really a pioneer in personalized cancer vaccines. He was really there from the beginning with the first neoantigen vaccine in melanoma in 2017. And was really instrumental in the kidney cancer vaccine. I remember the first days of the epitope selection board, hours spent selecting vaccine targets. And so it's really a pleasure to have you speak today on the future of cancer vaccines.
Thanks very much, David, and thanks for having me at this meeting. So I know that you have talked about vaccines in this meeting already, and you actually, I think you presented the RCC data already, right? So, but I just wanted to make and we have also talked about antigens here in this session, actually pretty extensively, but I just wanted to make the point, so what is actually the objective of a cancer vaccine as the one background slide? And I think it's, at Dana-Farber in the early development of these vaccines, we always felt very strongly that sort of addressing diversity by targeting more than just one antigen is going to be critical, and that this personal approach sort of ideally, because typically you can sort of customize that as a vaccine, and you're not stuck with the one antigen that you identified in you basically just focus on that one. And then of course the vaccine is supposed to hopefully generate new T-cell immune responses, T-cell responses against the tumor, and also diversify and expand the T-cell repertoire, and hopefully those T-cells are going to be durably expanded and also functional over time. And so just wanted to contrast that concept because there's a lot of talk in the field about like shared vaccines versus the personalized vaccines. And I think sort of on the left side here, if you wanted to target just one neoantigen, let's say you have a KRAS vaccine, you don't really address diversity, at least not in that sense. Of course, you can always say like that first immune response can trigger epitope spreading and so forth. But at least as a sort of shots, in terms of shots of goal, you basically just focus on that one antigen. And then, of course, like you also limit the patient population, right? So typically, because of HLA restriction, and not your whole patient population is going to have that driver mutation, you really can give that vaccine rather than like the personalized vaccine, but in theory, everybody's a candidate. It could be just 20 or 30%. One approach that Gritstone took, a company that developed personalized vaccine but also shared vaccine, was basically to predict and sort of focus on a whole bunch of driver mutations and then have one vaccine and give them to patients that had like either of any of these vaccines. The problem in that study was actually that there was really kind of unexpected immune dominance where they saw T-cell responses against P53 in basically the patient population where the majority really had KRAS mutations. So that's a potential problem. And then of course that stands in contrast to the personalized where you can really customize like what you want to put in there. So just two background slide of what we have been doing at Dana-Farber, and you've heard about the kidney cancer vaccine already, but sort of in that first study, the design of the vaccine is just like the kidney cancer vaccine. Is 20 neoantigen targeted, long peptide vaccine. The adjuvant is a poly-IC. And we saw these really nice, robust immune responses when we looked at those first melanoma, in this melanoma study, that we then actually did a follow-up study on and were able to show that these responses were durable for, you know, we looked at 2.5 to 4 years follow-up, and the majority of these immune responses were actually still there when we looked at the later time points. We also did a study in glioblastoma, actually saw vaccine-specific TCRs post-vaccine. That was sort of one of the, you know, big findings in that study, and then did further follow-up studies where we actually generated tetramers for some of the neoantigens, and then saw diversification of the TCR repertoire, and also the functionality of these vaccine-specific T cells kind of really went into the right direction from like a naive phenotype to a memory phenotype, and we also saw epitope spreading. And then we actually did a larger study that was not done at Dana-Farber, but used a very similar vaccine, where we went into melanoma, lung, and bladder cancer, and saw, you know, quite encouraging tumor responses, but the study was not randomized, and so it was not sort of for the investors of the company sort of enough to put this further. But I will still say that a randomized study is, of course, like the best way to show efficacy in a clinical sense. But in that study, we actually did see surrogates for vaccine-induced tumor responses. One of them was we actually saw path responses after the vaccine that was not seen with PD-1 inhibition, and we also saw epitope spreading, which is also a surrogate marker for anti-tumor activity. I just wanted to talk about two prominent studies that really have led to a lot of enthusiasm, a lot of investment actually in the field of personalized neoantigen vaccine. One is a study by Vinod Balachandran, who did a study in pancreatic cancer in the adjuvant setting, where they resected those patients, predicted neoantigens, treated with 1 dose of atezolizumab, and then used the BioNTech personalized vaccine that actually targets also 20 neoantigens. And then the patients get chemotherapy. And sort of the main finding on that study was quite stunning, is that they saw that they used two ways to assess the immune responses. One was ex vivo interferon gamma ELISpot, and the other one was to look at TCR clone expansion by their clone track sort of approach to like determining from bulk TCR sequencing whether clones expand. And what you can see here on the left side is essentially like when you look at the orange part of those gray bars is basically the proportion of vaccine epitopes that induced immune responses. So I will say, so you can see two things. One, you have 8 patients on the left side that actually had a response, and then there's 8 patients on the right side that didn't, but there's also a lot of gray, right? So there's a lot of like vaccine epitopes that were included in this vaccine that actually ultimately didn't generate an ex vivo response. And then you see the same dichotomy when you look at the clones, where the 8 patients that had the ex vivo ELISpot responses also had clonal expansion. There's just 1 patient who had clonal expansion in that other group. And then really stunningly, they had saw this amazing like separation of the recurrence-free survival curves, where it's really like, it's basically if there was an immune response, there was no recurrences. They had a long follow-up paper that they published earlier this year, would actually, the curves not shown here, there's like actually 2 recurrences, but it still maintained that separation. And they used the clone track approach and actually showed this amazing durability of these TCR clones, where they were able to look at these sort of the contraction rate of the TCR clones, and they predicted that some of these clones actually like could last for like 100 years, sort of outliving the host, which is the main claim of that paper. And then, of course, we have the melanoma study where patients received pembrolizumab alone, which is the standard of care for a stage 3 melanoma, versus like the pembrolizumab plus the vaccine. And we saw this really remarkable separation of curves where the difference in the absolute in the risk reduction for recurrence is actually similar to the PD-1 inhibition versus like the placebo. So the hazard ratio is like in the 0.56, which is quite remarkable and actually maintained over time. And that led to like a whole slew of studies, one of them being an RCC, a phase 2 study where you guys are like obviously leading this, and I'm not sure where it is like in enrollment, but probably advanced, I'd imagine, right?
Yeah, finished.
Finished, okay. And the melanoma study is also, it's finished. The melanoma study is a phase 3 study, and we expect results in like pretty much like a year, roughly. And so So what do we know now? Where do we have data and where do we expect data? I just wanted to visualize this here. So when you look in terms of the patient numbers in dark blue, you see like the data, the patients where we already have data. And then in pink, you see like what we will have data of in the coming years. And so what can we do to improve? There's obviously a lot of ways to improve these vaccines. I think my big worry is like some of these like pivotal trials that are ongoing. If let's say the melanoma study is negative, there may be like really people are enthusiastic now, but they lose their enthusiasm oftentimes pretty quickly, particularly like in industry. But I would say like as a field, so vaccines, this is not just one drug, right? So it's really like, I think we have enough data now telling us that like this can and really generate much better immune responses, but also clinical responses. And we can like improve a lot. And so one example is just a study that we published just a week ago, actually, where we have a very small study, 10 patients. We added Montanide and ipilimumab to that initial formulation and see much better immune responses. So basically, rather than 20% of the epitopes inducing ex vivo responses, we see 60%. Montanide is a fairly old immune adjuvant, but there certainly speaks to the fact that like if you improve adjuvant, you can get potentially a lot more mileage. So we also saw like actually a lot of like TCRs that were, proportion of them were specific for the vaccines that came up specifically after the vaccine that were distinct from the ones that came up after PD-1 inhibition. We can certainly improve our prediction algorithms. I'm not going to go into any detail here, but like what we're doing right now is essentially looking mainly at RNA expression and the binding, but we can look at the TCR side of things. We can actually become better in like discriminating between self and potential like pathogen responses and so forth. There's a lot of like room for improvement in that sense. And we already talked about the neoantigens that are not encoded by what we can currently like detect, it's basically SNFs and indels. We talked a lot about the ERVs, and then of course transcriptional and translational alterations. We're basically currently blind to, but there's a lot of opportunity to use those as neoantigens as well. I think there's also a lot of room for better engineering, even on the RNA side. So when you think about the RNA vaccines are actually, they need like formulation nanoparticles. Otherwise, you couldn't even do it. The naked RNA vaccines are not very effective. But of course, you can also engineer more into the RNA other than just the antigen. For example, there was a study by Drew Weissman earlier this year, just where he basically added like another IL-12 RNA that really enhanced the immunity after the vaccine quite a bit. And actually the same thing is true for peptides, right? So right now, we give the peptides just as soluble peptides, but you can actually do like nano technology as well. You can use nanodisc, and there's already data in mice and non-human primates that it can really generate much better immune responses when you do that. But that's actually not in the clinic yet. And then just like sort of as a shout out, as like, I feel like there's a lot of enthusiasm now that these vaccines are probably better in the adjuvant setting, but I would still say as like a combination therapy, if we have the right tools to address the TME, they could also actually be, I have not given up as vaccine potentially be helpful in the metastatic setting as well. And stop here, and I'm happy to take questions.
Susan.
Question patients often ask, and it was what you were addressing at the end, is using vaccines in the advanced disease setting, how might that develop?
Well, we have done studies in the advanced setting, and there's definitely in melanoma, there's data that tell us from clinical trials actually that going into like a third, fourth line setting is probably not the right way to do it. And there's actually even a first line study where we haven't seen the data, but there was at least a press release where if you do vaccine plus pembro in first line metastatic melanoma, it doesn't seem like there was the same sort of a similar difference from sort of compared to that adjuvant setting. And that really, that's what I mentioned, like, well, that's why everybody is like basically now saying like, well, they're only gonna work in the adjuvant setting. My point was more, well, why is that? And how I think we still don't have, and that's just a broader issue for the field of immunotherapy, right? That we just don't have the right tools to address like adequately the tumor microenvironment.
Yeah, so with regard to that topic, in melanoma, we saw in stage 3 in the neoadjuvant setting about the same benefit over adjuvant therapy with single agent anti-PD-1 as you see with adding vaccines. So that suggested to us that the issue was having enough T cells in the tumor so that you could actually activate them more against the tumor than, let's say, against the colon. Do you think that that is applying to these vaccines? You think that applies to kidney cancer? And is that why the adjuvant setting is a better setting?
So you're saying that like, so the PD-1 inhibition works better when the tumor is present, and that could be because like the TILs are in the tumor, right?
Right, more TILs in the tumor than in your colon, let's
Yeah.
say.
But I think it also comes to, I feel like the vaccine is separate from the neoadjuvant. And we are actually, so we're thinking of like using the vaccine for the non-path responders after neoadjuvant, right? So because the thinking is that there's like, this is not necessarily overlapping. Like the pool of T cells that you engage with that PD-1 when a tumor is present, right, may be very different from what you can actually generate with a vaccine. So I think it would be very like reasonable to try that, right? To basically say like, okay, that approach with the PD-1 inhibition plus with the tumor there, which basically the neoadjuvant has not worked in that patient, but maybe if we like actually generate new T cells against them, that could actually potentially be effective for those patients.
So a second question related to kidney cancer is, in contrast to maybe other tumors, in many patients only a small percentage of the primary actually is responsible for the metastasis, and maybe the neoantigens that might be present in the metastasis might be different than what's in the primary. So do you think that that's actually true? Have you looked at that? And would it be better to make vaccines from oligometastatic lesion rather than from the primary?
I mean, with that rationale, certainly, right? I mean, in our CC study, we had 2 patients, right, where we got like, we actually generate.
So there were 2 patients that had oligometastatic disease on the study, so we did end up sequencing both the primary and the met, and I think our results actually mirror what Samra had seen in TRACER-X, which is the vast majority of those mutations were not private to the metastatic site. So over 80% were shared. The clonal frequency might have been different, so something might have been subclonal in the primary and clonal in the metastatic site. But I think about 80% of the vaccine targets ended up being shared between the primary and the met.
Great, thanks.
Yeah.
So it was very interesting at the end you mentioned nanodiscs, and you're probably aware that in industry, nanodiscs are being used to generate antibody responses, right? To hard to treat targets, like you can send to a CRO and get a nanodisc experiment done. So I was wondering if you think you're going to also be able to, the epitope is probably small, but have you ever looked at any of your studies, or can you look to see if you can develop antibody responses to your targeted neoantigens?
So that's actually a great, so we have not in the melanoma study. I know that Nina Bodwatch has looked at antibodies pretty extensively in her studies. She definitely sees antibody response. I don't know the data in detail enough that I could tell you like what exactly but it was certainly like she has seen antibody responses.
I just think when you move to nanodiscs, you might be more likely to see it, you know what I'm saying?
Yeah. But so the mouse data that they're actually by now like 80 years older. So like Dave Moon from like Michigan, he basically had these like mouse models, and this is all T cells, the CD8 responses, and actually neoantigens. So the data in mice looks great. So where you think like, oh geez, we gotta use that, but I'm not really sure. Actually, speaking of industry, what is actually the impediment? Like, why is this not being developed for? I don't know.
So maybe a more general question for the panel, which is, just going off of Dave McDermott's talk earlier, if you believe that a cure is possible in RCC, I suspect immunotherapies are going to be the way to get there. But often they're tested in, as Patrick said, very late line settings, which is really tough. And so just thinking about kind of the clinical development path, what do you think is the right strategy for developing a lot of these novel immunotherapies in terms of which line of therapy, in terms of a single agent versus co-therapies? How do you sort of think about the development of these? Can maybe go down the path, yeah.
So I think one thing that drug companies are famous for is stepping, right? So basically you start in the latest stage and then you go to like the second latest stage, then you go to the third early. And I think that the way to do this actually is to try to jump. I mean, once you have safety and efficacy in late stage, very late stage disease, trying to move to that earlier stage. The very big challenge is that there's standard of care, which works. And so, you have to take a risk and patients have to take a risk. But I think you're not going to get there unless you take that risk.
With that, I think once you have safety data on one of these cellular products, I think you should not insist maybe on those later stages and try to move fast to earlier stages. At least some of the protocols on cellular therapy, they also give in the eligibility criteria that language, patients might have failed these therapies unless they decline. Because I think the patients also need to have this autonomy to decide what they want to be treated with. So I think this is my experience, at least with the protocols that I have worked with.
Tony?
So I think since the publication, David, with you of our Nature paper, we have 2 to 3 requests a week to get on the vaccine. And we finished the Merck study, but even in patient late stage, we have to think about a strategy. We're seeing, yes, the vaccine is earlier, the PD-1 earlier. I'm not sure how that it's true. That perhaps is true if the vaccine is given as a single agent. What about a strategy to, completely downstage the tumor or treat it with a combination that has a TKI? A lot of these TKIs have immunomodulatory activities, plus PD-1, and then giving the vaccine as maintenance in late stage. I mean, that could be a right strategy. For clinical development, I like it because it buys you time. You can't make the vaccine overnight. You need, several weeks. So you can get those patients, give them the TKI, a TKI, for example, like cabozantinib, that has immunomodulatory activities, wait, get scans at 6 weeks, make sure at least you have stable disease. By that time, you hit with the vaccine, with or without the TKI at that time. That's the clinical development. You don't deny those end stage, those advanced patients, the potential for a strategy that might be active and certainly will preserve quality of life.
Yeah, absolutely. And I think, the adjuvant therapy has been setting has been appealing so far, both because you have time, but also because it's that minimal disease setting. But I think if you have what Patrick sort of hinted at the end, if you have the right co-therapies, I think being able to buy time and actually administer something that's going to be effective, not as a monotherapy, but together with other approaches might be really something.
And I think it really gets to study design, and we just have to be a little bit more innovative in how we look at the endpoints, right? So ctDNA is everybody mentions ctDNA, but I will say ctDNA actually has been used, for example, in the Gridstone program, and they maybe have put a little bit too much attention, I would say, because basically, you look at the ctDNA plots and it looks like rhesus, and it's there's a correlation if you have a ctDNA response and with survival, and how much does that mean? But you could certainly envision that could be at least a layered on endpoint of a study. And I think when you think about metastatic disease, and what most patients want is durable responses, right? So, and I think with the vaccine, we really have to, that's different from a drug that can give you an immediate response. And we know that from the melanoma adjuvant, the Moderna study, I think when you look at the survival curves, right, you see that they don't separate for 6 months, right? So it would be, you can tell that it takes some time for that immune response to build up and then generate actually But I think we have to have a smart way to sort of layer that on in the design, and actually then focus, let's say, even in the adjuvant setting, right? You could focus a little bit better on like the high-risk patients and do the segregate the studies or stratify in the studies, or even just give vaccines just to the patients where you think they need them. But also in the metastatic setting, right? You could like just be a little bit better in teasing out who the responders are. Even I would say like in that NEON study that I mentioned, right? So that was like, I sort of alluded to the fact that like people were like, oh, I know it's not 80% response rate, and we didn't see. So initially when we actually designed the study was we treated the patient with PD-1 inhibition and then gave vaccine like 3 months later, just because obviously you gotta give something while they make the vaccine, so it was like a natural approach. But the thought was like you have like the patients as their own controls, right? So you could have like, let's say 40% response conversion, as we like termed it at the time, right? But that when we didn't see that, people were like, oh, the vaccine is not doing anything. But there's definitely like, there's clearly some patients that were treated on these trials that benefited. It's just like harder to tease out who they are and like sort of convince people that this is actually real.
That's great. Well, this has been a wonderful session. I want to thank all the speakers. It really is, think about the next generation of 1, 3, 5 years to come for immunotherapy and kidney cancer. And so before we break for lunch, just a few quick words from Brian, but thank you again to our speakers.
It's been a great morning, some wonderful conversations, a lot of dialogue, a lot of collaboration, love it. We're gonna have a great lunch right outside there, but I hope you get outside for at least a minute. It's glorious outside. Enjoy the afternoon sun, but get back in here at 1:15. So it's break time, lunchtime. Thank you, everybody. Great morning.